lot. Okay. You can't see it up there? You sure can't. Thank you. Okay. So, we're talking about genetic material. The genetic material, as you guys know, these are nucleic acids. This is DNA and RNA. So what the deal is with DNA is this is where the instructions are. They're coded within this molecule, within this double helix. Here's a very famous model of DNA. Now you guys should know that these monomers are called what? The monomers, so the building blocks are called nucleic acids, okay? And they're made of 3 parts. You have a phosphate. Do you remember the orange pentagon? Sugar, deoxyribose. And the inside is one of the, it's held together by hydrogen bonds, a nitrogenous base. Right? So the nucleotide, I'm sorry, I said nucleic acid earlier. The whole thing is, or a chunk of it's called a nucleic acid. The monomer, the component, is the nucleotide. Okay? So you have your nitrogen-containing base, your sugar, and a phosphate group. And then the 2 bases, because you have another nucleotide over here, are held together with what? Hydrogen bonds. Now, the phosphate to sugar, sugar to nitrogen-containing base, those are held together strongly with what? What type of bond? Covalent bonds. Now, the hydrogen bonds are holding these so they can easily unzip apart. You guys remember that for DNA replication? Okay? So, that was replication. Now we're moving on to a new process here of using the code to make the proteins. Okay? Okay, so again, we're about making proteins. Now, what has to happen is there has to be a reason that we need a certain protein made. And the cell will send signals. Maybe it's red blood cells, and they need more hemoglobin. They need that protein that's going to attract the oxygen so they can carry the oxygen. So they need, with the body senses, hey, we need more of this. So then that is coded by a gene on a chunk, right, of DNA in a specific chromosome in the cells. So what happens is the enzyme makes a copy of the code from the DNA. So just like with DNA replication, the DNA has to unzip, and it just reads one side of the DNA to make actually what we call messenger RNA, which is a single strand. And messenger, it's going to take the code because the DNA is stuck where? In the nucleus. But we need to get the code to the ribosomes. Remember the ribosomes? They make the what? Proteins. So that's where it'll be translated. So we say that the DNA is transcribed into the messenger RNA. The messenger RNA leaves, goes to the ribosomes where it's translated into a string of amino acids, which amino acids are monomers of proteins, of making our big protein. You remember catalase? Remember the enzyme catalase? Remember that big globular picture I showed you? That's a protein. So we need to make the protein. All right, so that's in a synopsis there. I mentioned the messenger RNA. It's a temporary copy. It's a complementary copy of the DNA that is carrying the instructions for making the protein. And it can be used over and over, but eventually it's fragile and it'll break down and we'll just need a new copy. The process of making the messenger RNA is called what? Transcription. It's transcribing it. I don't know if you guys ever heard of a transcriber. There's what are those people called in court? They are transcribers, but they haven't met court reporters. Have you guys ever seen those court reporters? So they still use this old system. It's normally, not to sound gender, but I think most of the time it is a female, but it doesn't matter. A lady sitting over in the court, and she is typing really fast with a different code, and it's like there's different things, lines and different things, I don't even know how it works, equaled words. So it's a really fast way of typing with some symbols. Check it out if you're interested. Still used today. So she's transcribing. She's taking the words that she's hearing and she's putting it down on paper, right? So she's changing the code to a different form. Well, that's what's happening. We're taking the DNA code, we're transcribing into a different molecule that's carrying the code, though, the same thing, okay? Now, this transcription was obviously very important before video technology and tape recorders. It's been going on for a long time. But still, people like hard copies. Okay, then the translation. Now we're going to interpret the messenger RNA into the string of amino acids which will build the protein. Now, there's more RNA. I've mentioned one RNA. What did I say that one was called? Messenger RNA. Remember, in the ribosomes, there is also ribosomal RNA. And I'll talk more about RNA in a minute. Then there's also transfer RNA. And there's the three main RNAs you have to remember. There are other types of RNA that still help with the process, but they're still discovering them. We don't understand them as much. There's some RNA in the nucleolus, for example, in the nucleus. But we're just going to worry about these three types. Okay, so a nice synopsis. We have our DNA. We're going to make from the DNA messenger RNA. Now look at it. Look at the messenger RNA. Do you see how different it looks compared to the DNA? Now the messenger RNA moves to a ribosome. What color is the ribosome there? Yes, you guys see that? It actually fits in a certain spot, kind of like an enzyme, and it is read and translated, and you'll see, into the amino acid chain. We also call this polypeptides, which will form the protein. Okay, the code. Like, I don't know, that court recorder, they have their code. Life has its own code. We've just interpreted this, right? We didn't build this. This came with life. So it's 4 letters for DNA. And what it is the genetic code requires at least 20 different code words, one for each amino acid. There's approximately 20 different amino acids. Amino acids are monomers of what? Proteins, right? We string amino acids together, and that's what? A protein. You guys waking up for me? Okay? So there's 20 different building blocks. For the DNA, do you guys know the letters? A, T, C, and G. Doesn't matter the order. There's 4 different monomers here. How many amino acids are there? 20. So there's 20 different monomers for a protein. Okay? Patterns. You guys, if you guys will see these patterns, it'll be easier for you guys to tackle the tests. Okay. So, now just mathematically. There's 3 nucleotides. If we group 3 of them at a time, we can get 64 different triplets. Okay, if you think about zip codes, they used to be, all zip codes were just 5 letters. Not letters, 5 numbers, right? Out of 1 through, 0 through 9, right? A single digit. And we've made all these codes. Same thing with telephones, right? 7 numbers. So we only need 20 different codes. There's only 20 different amino acids. So a triplet is what the molecules did. And we have a possibility of 64, but we only need 20. So it's redundant. And that'll make more sense in a minute. Okay, now back to RNA. You need to remember that RNA is single-stranded versus DNA is what? Double-stranded. Okay? It's single-stranded. The messenger RNA is just a line, right? A line of the code. Transfer RNA is folded in a very specific shape to do its job. To bring over a what? Amino acid. This area of the transfer RNA is called an anticodon because it's the complement of the messenger RNA. Now there's one other big difference. RNA does not have T's. RNA does not have thymine. In place of thymine, it has a different one called uracil. Better get that down somewhere. Uracil. Now, scientists don't really know why RNA uses uracil instead of thymine. There are some hypotheses out there. The one that I think makes sense is just so the cell can quickly identify, hey, that's not a piece of DNA. The cell only has one copy of your DNA in the nucleus for protection. We do not want to be messing up DNA, right? RNA, no big deal. We can make more whenever we need it. So it makes sense to me. It's a hypothesis that it's so the cell could say, hey, that's not a chunk of DNA. Okay? Anyway, where it's folded are those complements. Okay, so the base pairing. In DNA, A always bonds to T. But if you're making RNA, it's going to be a U. C always to G. Okay, so look. C to G, U to A, C to G. So do you guys see how the transfer RNA is also reading the code, bringing the correct amino acid to the area? And linear, in a line, you'll see. You guys are doing great out there. Okay, scientists had to break the code and figure it out, okay? So check it out, everybody up here. Look, 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 everybody. Actually, how big is this in your paper? It's microscopic, right? It's straight out of your book. Colton, you with me? Okay, so you have to read the code. So, on the messenger RNA, not the anticodon. So let me just say it's GAC is the code. Eyes up here, you guys. Look how the box works. First base. What's the first base? G. So I'm going to put my finger down here. Second base, we're going to use the top. What's the second base? So I'm going to slide on over here. Now what it's doing, what it's coding for is one of those. Then what I got, C, third base, boom. That code will attract the transfer RNA that is carrying aspartate, one of the amino acids. Got it? Easy code, decoder? Question? I don't know. We're in chapter 9. Yeah, it's easy to find once you turn there. Okay, so remember, proteins. Are there a ton of them? Yeah. Do they do different things? Yeah. You remember that? Because protein's one of the 4 big molecules of life. So for example, we got keratin, collagen, myosin, thanks. Serve as the material for making up cell structure, tissue. I think keratin is in the plumage here. You have collagen in your fingernails. Enzymes like catalase, remember that in the liver, you guys, breaking down hydrogen peroxide. Proteins, hemoglobin, bonding, or actually just kind of attaching. Okay, so all the proteins that you need and that you're built of have to be coded by the DNA. Insulin. Okay, so hormones are proteins. So again, you have a gene that codes for it. This is where genetic disorders come up. Is there a mutation? Is there a problem and your protein is not being built right? If your protein's not being built right, it's probably not going to work right, and you're going to have problems. Okay? Diabetes is an issue with not getting enough insulin made. Okay? Okay, we're good on that. Okay, now, I went over the overalls. This is going to get a little more specific. So gene expression begins with RNA synthesis. I said that. Of course, there's an enzyme involved. What was the enzyme for DNA replication? What was the main enzyme that put the A's on the T's for DNA replication? Remember your models, your little pop beads? DNA polymerase, poly. It's just making many of these things. It's putting many things here. Here, of course, the enzyme's called RNA polymerase. Prokaryotes. What are prokaryotes? Bacteria. They only have one type of RNA polymerase. Eukaryotes, of course, were more complicated. They have 3 different types of enzymes for doing this. Protein synthesis takes place outside the nucleus, remember? However, messenger RNA, transfer RNA, and ribosomal RNA are all built inside the nucleus. The ribosome is made up of 2 parts called subunits. So a little more detail there. The code for making the transfer RNA is also just like we would make proteins, but now it's coding for making the RNA. Okay, I went over these pretty good here. You remember your 4 bases, except RNA does not have T. It has what instead? U. Okay, first for transcription, the first thing that needs to happen is called initiation. I don't really care about you memorizing this. I just want you to know the overall process. But of course, it's going to start. The enzyme RNA polymerase attaches to a specific part. We only want one part read, one gene read. It's called elongation when the RNA is being built. We will play with the pop beads again on this so you guys can see the difference between DNA replication and transcription. But just like before, the DNA is going to have to unzip, right, to open up the template for the RNA to be made, this time transcription. Then termination, it needs to stop at the right spot. And there's a code called a stop codon to say, hey, that's it. Go, messenger RNA. Go, help assemble your protein. Okay, let's check this out.
Transcription of RNA from a DNA template takes place in 3 stages. Initiation is the first stage. It occurs when the enzyme RNA polymerase attaches to a promoter region of the DNA. The promoter region is located just before the segment of the DNA coding strand that will be transcribed. The second stage is elongation of the RNA. RNA polymerase partially unwinds the DNA, exposing the coding strand of the gene. The enzyme moves along the DNA away from the promoter site as it builds an RNA molecule. A single complementary strand of RNA, called a primary transcript, is made. The third stage of transcription is termination. When RNA polymerase reaches the terminator region, or the end of the DNA to be transcribed, the enzyme and primary transcript are released from the DNA, ending transcription.
So you can see it happening right here. This is the DNA strand. It's separated. Now, different than replication, we're just reading one side. If we were to read the other side, it would go backwards. And just like our language, we can only read one direction. You guys got that? And you can see what's happening here. These black things, right here we have the, so this must be, yep, in bacteria. There's no nucleus, right? So you see it coming right off here. So this is E. coli. So we got DNA doing transcription, making messenger RNA. The messenger RNA, you can barely see, it's the strand. It's plugging in to little ribosomes. and that's where your proteins are going to be spitting out. So in eukaryotes, like I said, messenger RNA can be used over and over, from minutes to days, depending partly on how the primary transcript is processed. That'll make a little more sense. All right, so the code is obviously still very fragile, and it needs to be taken care of. So what this part is talking about is we put a G cap. We basically put a bunch of nucleotides to protect it. So it's got a cap on the end of what's going to be transcribed, and it even has a tail. Again, so here's the important part. These are protecting the pieces so that they don't break off. Okay? Now, also notice that, and this is pretty cool if you think about it, that there are parts of the code that need to be cut out. Now, the deal is one gene can make a messenger RNA, and that messenger RNA can be edited differently to actually make different proteins. This was a big revelation. When the Human Genome Project happened, scientists were very surprised that we didn't have that many genes. I think it was about 30,000 different genes, that's it, on our DNA. They thought there would be like hundreds of thousands of genes on our DNA. There's billions of base pairs, right? Well, then we realized, hey, there's only about 30,000 genes. What's the deal? Well, one gene, if it's edited differently, will code for different proteins. We pull this piece out, put it back together, that codes for that protein. This one over here, we're going to pull this one out and this long chunk out. Now there's that code for that. Does that make some sense? A little higher level there, not overly worrying about it. But what you do need to understand is it is edited. Now, I think it sounds backwards, okay? So introns are actually removed. Exons are actually put back together, and it's because it's like expressed. That's the information that's exiting to the ribosomes, is why they named them that way. Okay, so the exons are what's being expressed into the protons. The introns are in between, not important. They are spliced out and broken up and reused. Okay, I know it's going a while, but we need to get this done. Okay, just a little more, showing you a little more detail with what? What's that called? What type of RNA? Exactly, transfer. It's transferring the amino acid to the polypeptide to make the protein. Okay, now we move on to translation. So check this out. This is happening at the ribosomes, like I talked to you about the anticodon spot. This needs energy. Of course, we're going to use ATPs. So look at it up here. What is this, everyone? What type of RNA? Messenger RNA. What are these? Transfer RNAs. Look at this one's going to match up perfectly to the code. When it pops over here and bonds, this will be close to that, and that'll bond. When this bonds to that, it breaks off the transfer. This is an empty transfer RNA. It'll move off. It is attracted to its amino acid. It'll go find its amino acid, get like recharged, and it'll be used again. So that was just a picture showing, here's your growing polypeptide chain, here's your messenger RNA, like a typewriter, boom, 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 or train, okay, moving through. So you can see the direction of the movement. Just like I talked to you guys about on the last picture. And here's going to go over the whole process. Again, there's a stop codon. There's actually, I think, 3 different ones. So here's the complete overview. So to give you an idea, to show your understanding on the exam, if I took the words off there, you should be able to identify every piece of that and tell me what is occurring. Okay? So, got transcription over here, you got translation over here, for example, and all the different components. That is how proteins are made from the DNA. Okay, let's check this out.
Proteins that will become part of membranes or leave the cell are made of ribosomes that are bound to the endoplasmic reticulum, or ER. The first few amino acids of these proteins are the signal sequence that directs the ribosomes making them to the ER. The signal sequence binds to a receptor protein in the ER membrane. As translation continues, the growing chain of amino acids threads through the membrane and into the space inside the ER. When translation is complete, the signal sequence is cleaved, and the new protein is released from the ribosome into the inner ER. Often a sugar molecule is added to the protein, forming a glycoprotein.
So you remember before, what's ER? Endoplasmic reticulum. One of their functions is to what? Their main functions. Modify proteins. So this is what's happening right here. Proteins need sugar added to them, might need a lipid added to them, might need to be folded better. So remember attached ribosomes? That's called rough ER, right? So that's what it's showing you. There's that attached ribosome right on the endoplasmic reticulum membrane, right? So very efficient by its location. Okay, so sometimes there are mistakes. It's very few. Typically when DNA is copying itself, one base is placed improperly out of a million. So that's a DNA mutation, but also now here reading it, making the messenger RNA, you could also have the wrong base put. And then it might mess up the code. Remember I said the code is redundant, so it might not. So, different types of mutations here. A frame shift is one of the worst because it messes up the whole sentence. So eyes up here, you guys check it out. We have AAU being the codon. Here's the amino acid it codes for. You shift one frame. Then all the three codons are read incorrectly and you're building something that's not going to work. It's not anything. Okay? Now, this totally reminds me of if you guys skip a space on your Scantron and you don't go back and check your numbers. They're all in the wrong spot. Or you bubbled two answers on one. You pulled it back the other way. So a deletion or an insertion is going to do a frame shift because the codon's read in 3. Does that make sense? Okay, so we are going to stop after this couple little review items. I don't think that's on there. I want you guys to do this right now. So write it down. How will the complementary strand of RNA be coded if the DNA is coded? So right now, write down your messenger RNA strand. Quickly. Just put it anywhere. I don't care. If you could see it, you could see it. So I want you to make messenger RNA. Okay, so I'll start you. What goes to a G? I'm going to go ahead and write messenger RNA so I know what I got here. C, what goes to a C? What goes to a C? G. What goes to a T? A. Keep going. I would also separate them. See how I spaced them out?